The overall goal of this procedure is to determine the location and structure of calcification event by performing a combination of optical and electron microscopy methods. This is accomplished by monitoring the living larval tubeworm during metamorphosis, the life stage responsible for calcification that can be detected using fluorescent indicators sensitive to intracellular pH and calcium signals. Intracellular pH imaging allows us to study the concentration of proton into and out of the cytoplasm in the process of calcification. To begin intracellular pH imaging in the marine tubeworm larvae, the competent swimming larvae are stained with the intracellular pH indicator dye SNARF1-AM. The larvae are then transferred to a dish of IBMX containing seawater with a thin glass observing window and placed on a fluorescent microscope, where they are hit with light at 488 nanometers in wavelength, which is absorbed by the dye. 580 nanometer and 640 nanometer emissions from the dye are collected. The values of intracellular pH can be calculated from the ratios of these values. Optical microscopy allows us to identify time and tissue region associated with the higher pH level. This is the first step to determine the time points of interest for further analysis. In the second step, preserve the tubeworm with fixatives at the relevant time points for electron microscopy. A newly mineralizing life stage is identified using SEM-EDS mapping for calcium signal. Then, the regions with a high calcium signal are screened using SEM-EBSD, identifying the presence of crystalline mineral. Finally, using a focused ion beam, the mineral is cut out. A thin section is prepared for TEM.